Hey guys, we're going to go ahead and talk about Chapter 8, Part 2, Enzymes and Energy. Make sure you're filling out your Cornell notes. All right, let's go ahead and move on. So what is metabolism? Well, metabolism is all the chemical reactions that keep an organism alive. Some examples are digesting food, breaking down glucose to produce ATP, and using ATP to provide energy for protein synthesis. Can you think of any other examples? Maybe you can use this picture as one. We'll talk about some other examples on Tuesday as well. So let's talk about a metabolic pathway. A metabolic pathway begins with an initial substrate. It has multiple steps which are catalyzed by different enzymes. It ends with a final product. So for example, A turns into B, B turns into C, C turns into D. Enzyme 1 helps with this reaction. Enzyme 2 helps with reaction 2. Enzyme 3 helps with reaction, what was the third reaction? So how could a cell produce more product? Or produce less product? Or shut down this pathway? What would happen if product C, for example, let's just say product C was inhibited the activity of enzyme 1. Well, if product C shut down enzyme 1, then, sorry about that, guys. So if we have C shut down enzyme 1, once enzyme 1 shuts down, that basically means that B will not be made as fast, which means that it may hinder the progress of making C and D. Okay, so here's the thing. Cells can regulate control metabolic pathways by expressing or repressing genes that encode enzymes in the pathway. They can activate or deactivate enzymes in the pathway as well. So what is the initial substrate in this pathway? What is the initial one? If you point it to the top as threonine, you are correct. What's our final product? What is our final product? Follow the arrows. Our final product is isoleucine. So what does the final product, or how does it affect enzyme 1? Well, let's follow the arrows. What it does is it goes and it binds to the enzyme, so now nothing can bind with it anymore. Until it releases, then the active site is available and we can do it again. So for a moment, it stops anything else from happening. So in feedback inhibition, the final product of a metabolic pathway shuts down the pathway. Feedback inhibition prevents the cell from wasting energy and resources. So is this an example of a negative or positive feedback? We learned about some similar pathways when we were talking about Chapter 1. So are we stopping things? Are we adding more? Well, if you look at this, we're actually stopping this enzyme from working more, so therefore it will be considered negative feedback. So is this an example of specific binding? Does it look like it will fit specifically? The answer is, well, yes. Enzymes are proteins made of amino acids. What would happen if a mutation caused the active site to have a polar side chain where it usually has a non-polar? Perhaps if this side ended up having polar where it was supposed to have non-polar, perhaps it will not fit anymore. Does this enzyme change shape at all after the substrate is bound to it? Well, if you look, there's more of an opening right here versus over here, there's a very tiny spot. So does it change shape? Yes, it does. So why is the shape of the active site important? Well, substrates bind specifically to active sites. If the shape of the active site changes, the function of the enzyme also changes. So let's talk about induced fit. Induced fit is when a substrate binds and the enzyme changes shape just a little bit. It doesn't change crazily shape, just a little bit, so that it fits even more tightly around the substrate. So let's go ahead and take a look at this diagram. I know you have this in your paper, so let's fill it out. So in step 1, the substrate enters the active site. Step 2, the substrates are held in active sites by weak interactions. In step 3, the active site can lower the activation energy and speed up the reaction. Step 4, the substrates are actually converted into products. So now we actually went from reactants to products, which is why we have a different color. And then step 5, the products are released And now the active site is actually available, so we can actually do the whole thing again. Make sure you go over this process. So how do active sites lower the activation energy barrier? Well, the active site of an enzyme can lower an activation barrier by positioning substrates next to each other, straining substrate bonds, or providing favorable microenvironment. Let's look at this. At what temperature do these enzyme catalyst reactions, these enzyme catalyst reactions are the fastest? So this is the rate of reaction, so we're looking for the highest points. So if I looked at this optimal temperature, it would be at the top of this peak, which is 37. Or the peak of this one would be at 77. It says it right here too, guys. So in case you're wondering where am I getting those numbers. And over here, 37 and 77. At what pH do these enzyme catalyzed reactions, which ones are they, where are they the fastest? So this is the rate of reaction. Wherever it's the highest is where it's the fastest. Here's our peak at about 2. And here's our peak for trypsin, which is in the intestines, which is at about 8. So environmental factors can actually affect enzyme activity. So enzyme activity can be affected by temperature, pH, substrate concentration, and presence of inhibitors, activators, and cofactors. Each enzyme has an optimal temperature and an optimal pH, which favor its most active shape. So how do inhibitors affect enzyme activity? As you can see, here's a non-competitive inhibitor, and the active site has actually changed. We have competitor inhibitors, which bind to the active site, and they block the substrate from binding. They compete with the active site. So here's an example of a competitive inhibitor. Now this little guy can't even fit into the active site. We have non-competitive inhibitors that will go ahead and change the active site. And so examples of these are like toxins or poisons or antibiotics and pesticides. They'll go ahead and do that. That's why you have to be careful that you don't get it into like your eyes or or you accidentally digest it. So how many subunits does this enzyme have? Well, let's count. 1, 2, 3, 4. How many active sites does it have? Well, it says active site of 4, so 1, 2, 3, 4. The enzyme oscillates, so it goes back and forth between what two forms? It has the active form where it's open and ready to bind, and then it has the inactive form where it's not ready to bind. So when we say oscillate, it's like swinging back and forth between the two. So how does this allosteric activator affect the enzyme? So we can see that we have an activator. Once we put the activator, it actually opens it up. But when we put an inhibitor, it's going to go ahead and change the active site so nothing can bind. So make sure that you summarize how these two will affect enzymes. So what is allosteric regulation and how do they affect enzyme function? Well, allosteric regulation is an activator or inhibitor that binds to one part of an enzyme and affects the shape of other parts of the enzyme. The binding of an activator stabilizes the active form of the enzyme. The binding of an inhibitor stabilizes the inactive form of the enzyme. Let's talk about cooperativity. Cooperativity, another type of allosteric activation. So cooperativity is a type of allosteric activation. So when one substrate binds, it actually stabilizes the active site in other parts of the enzyme. So for example, if it binds right here, all the others open up. What is the structure and function of cofactors? Well, cofactors are non-protein molecules or ions required for enzymes to function. They can be inorganic or organic. So an example of like a coenzyme would be like vitamins, for example, or like vitamin D from the sun. Where are enzymes located in the cell? Well, some enzymes are concentrated in specific organelles. For example, enzymes for cellular respiration are located in the mitochondria. And that's it for today. Make sure that you go over the notes and make sure you do your summary for the first one. And that's it. That's about it. You guys have a great day. Enjoy the weekend.